Dr. Collins, we're about to start the webinar, just so you know.
He organized it. He has got them. Oh, no. No, I didn't do that. No, I had a nice dinner. Where'd he go?
Good morning. Come on in. Come on in, we'll get started in just a second. Good morning. Come on in, anyone who's just joining us. Plenty of room for a celebration like this. Come on in. Hi, Kate. Good morning, everybody. Welcome to UMass Chan Medical School. What an amazing day for the state's only public medical school. That's in spite of the weather outside. Mother Nature didn't seem to agree with us. So in just a minute, we are going to hear from the man of the day, Victor Ambros, PhD. He has been on the faculty here at UMass Chan 15 years. 15 years, woo! For the journalists in the room who've joined us, thank you. Or if you're online, you will be able to ask your questions, hear from the laureate directly about his work. UMass Chan's first Nobel laureate, Dr. Craig Mello, is also here with us today. Where'd you go, Craig? Great. I should say my name is Jennifer Berryman. I'm the Chief Communications Officer here, and so thrilled that all of our years preparing for this is finally paying off. This is so great. So if the technology cooperates, we're going to be joined first briefly to hear some remarks from the Chief Executive of UMass Chan Medical School, Chancellor Michael F. Collins. If you're able to bring him up, I'll feel a little bit better. Chancellor Collins, hopefully you can hear us. The Chancellor is celebrating from afar today with all of us in spirit. Excuse me. Just so you know, the Chancellor is actually receiving an honorary degree from the University of Crete. And thus, he is unable to be with here physically, but I know, as all of you do, that he is probably the biggest cheerleader and ambassador for this medical school anywhere he goes in the world. And Chancellor, if you can hear us, please feel free to share your thoughts. Chancellor Collins, can you hear us? 1, 2, 3. Got a feedback loop. Okay. Let me text him. He does, yeah. Chancellor, can you hear us now?
Yes. I'm so excited to offer my congratulations to Dr. Ambros, Victor, congratulations on this real wonderful accomplishment. I'm very disappointed I can't be with you, but I am in Greece with Dr. Michael Czech, who was the chair of the department when you were recruited to UMass Chan Medical School, and I want to congratulate you today on this really marvelous accomplishment. I was up at the time of the announcement, obviously, because the time difference, and screamed in the car when I heard the Karolinska make the announcement. So congratulations to you. Let me make a special note of congratulations to Rosalind Candy Lee, Dr. Ambros's wife, and very close scientific collaborator for these many years. Candy, I know it's been so encouraging for Victor to have you by his side as he's made these very important scientific discoveries, and congratulations to the both of you on this day. From an institutional perspective, I can remember the very first conversation I had with Dr. Ambros when we were talking to him about joining UMass Chan Medical School. And it went something like this. I said, you're very accomplished. You've won lots of prizes. Why would you want to come to UMass Chan Medical School? And his answer was very simple. He said, Michael, I feel a responsibility to come to UMass Chan because there exists the finest RNA biology community in the world. And for us to come together to make important scientific discoveries is something that I just feel would be the right thing to do and a responsibility. And Victor, I remember you making that comment and how excited we were to have you join our community. Let me thank Dr. Michael Czech, who was the chair of the department when you were recruited. Let me thank Craig Mello, who was instrumental in bringing Victor to our campus, and to the current chair of the department, Dr. Roger Davis, who has provided so much support to Victor. Tomorrow, Dr. Ambros moves into his new lab in our new education and research building, a lab that we've created so that he and the colleagues will be in there, continue to make important discoveries that will change the course of history of disease. Let me thank the Board of Trustees and the president of the university, Marty Meehan, for the resources that they've provided the medical school so that we could have such outstanding facilities and recruit such outstanding scientists. Victor, it's a momentous day for you, but I want you to know that the great pride that our institution has in you, what you've accomplished in science. So I think it makes so important the note that science for science discovery sometimes is the most important thing. And for us to have that tremendous support that we've received from the NIH and from other funding agencies so that you could do this important work is really a wonderful thing. Congratulations to you. I look forward to being able to congratulate you in person over the next few days. Thank you very much.
Terrific. Thank you, Chancellor Collins. He's going to stay on and listen to everything that you have to say. So let's not waste any more time. Come on up, Victor Ambros. 2024 Nobel laureate in medicine or physiology. Great news. I don't know if you want to just make some remarks. If anyone has questions, I'll just repeat them into the microphone for the purposes of those watching online.
I'd like to make a few remarks. I have no idea what I'm going to say, so it's like saying, all right, I'll take that barrel over Niagara Falls. But I know there's certain things that are important for us to try to emphasize in circumstances like this, and Chancellor Collins mentioned a few of them. One is the importance of the institution in which you work and the colleagues that you have and the leadership that's in place in that institution. I've never really appreciated it so much until I got to UMass, because here I discovered that institutions can really work and that leadership really, really matters. And the leadership of Chancellor Collins and Terry Flott, as a provost and dean of the medical school, to me, has been absolutely amazing. This medical school has, on this campus, probably more C. elegans and Drosophila laboratories than any other university. And it's no accident because, in part because of Craig's work, and in large part because the leadership has recognized from the beginning that model organisms are absolutely the drivers of new knowledge in the life sciences. And that hasn't really changed. The work we did, that Candy and I did years ago, and the Ruvkin lab did, was 30 years ago, but the same principle applies today. Up and down our corridors here, there's people discovering day by day, new surprises in life sciences. And those surprises are telling us that there's fundamental principles of living things that we don't understand, right? And that's today, and it's going to be tomorrow, and it's going to be for decades and decades to come. So support of the NIH has been absolutely constant for me throughout my career, and I often, whenever I renew my grants, I think, are they going to buy this again? And they do. The other thing, and Candy has given me a list of things I'm not supposed to forget. I probably forgot them all. I think the other thing that's so important, and so by the way, UMass, I want to say again, this is an institution that is, I think, getting right, as best as any institution can, any human institution can, the formula for making it possible for people of all sorts to come together and really do science in a way that's synergistic, where communication and sharing of ideas is the expectation, not the exception, where collaborations occur naturally. All of us in the room who are PIs have experienced students in our lab finding a student in another lab, setting up a collaboration, and then coming to the PI to talk about the collaboration, right? And that we encourage, And I don't know of any other institution where that's the kind of culture that is actively promoted. And the leadership here is really important. Alan Jacobson, one of the founding members of the community here, the founding RNA biologist here, and Michael Check. These are people who you can see they have been promoting this ethic and this way of approaching science, actively, and creating a community where this kind of thing happens. I think that's super important. That's what one of the things I wanted to emphasize is that leadership matters. Institutions can work really, really well. The other thing is I want to, point out is that, all of us in this room, I think, can probably say to ourselves, how come I'm here? Think about my grandparents, the journeys my parents took, and people before that. I'm a son of an immigrant from Poland. Candy's a daughter of immigrants from China. And in these stories, there's heroes that you hear about in our history that are everyday heroes who's brought us here, right? So I'm standing here, the science got done, Candy and I got together. That's because, of the legacy of the folks who came before us, I wish we wish, Candy and I wish that our parents could be here to see this. My dad was a war refugee in World War II, and he's from Poland, and he was a beneficiary of affirmative action. In Poland in the 30s, there was a program set up by idealistic educators, young people, who set up a program to raise money to support the higher education of poor kids from the villages. That was the goal. And they identified poor kids in the villages. And my dad's village had about 30, 40 people in it. And his teacher identified Longan Ambros as a talented individual. And at that time, you only went to the 3rd grade. And in the neighboring town, you could go, the law said you had to go to 6th grade, but they repeated the 3rd grade for 3 rounds. That was the education of young people in the countryside in Poland. So he got to go to, gymnasium in Vilnius, for a couple of years until the war started. And then he was homeless for a year and a half. And then he was captured by the Nazis, and he worked as a slave laborer, in a factory for a few years. And then he befriended, he was liberated and then befriended some people in the occupying U.S. Army, one of whom became his very close friend and sponsored his immigration to the States. And so that's how I'm here. His friend sponsored him, So I think that's really important for us to reflect on how, here we are on the, what was the other thing, Candy, that I was supposed to talk about? Yeah, public funding of research, We should all, okay, two levels. There's the state. This is a great state university, right? And I wish everybody who lived in this state knew how they can take proud ownership in what happens here, right? I'm sure if they knew, they would be proud, right? So I think that's first of all. And I think the public funding for research, as I said, from the NIH, Institute of General Medicine, supported me all my career. This is absolutely vital. And it's been said over and over again, and I think on occasions like this, we should once again say it. The taxpayers of the United States should rightfully feel proud of what they do through the NIH,
Maybe on that note, could you talk a little bit about your work that's being recognized with this award, and particularly for the non-scientists in the room who may not know?
Oh, that, yes, right, right. Yeah, so what we did, it was a team of people working in my lab, Rosalind Lee and Rhonda Feinbaum and myself, and our goal was to identify what was the gene product, what was the molecule or molecules produced from a particular gene. Now, we knew there was a gene there because there was a mutation in this worm. And the mutation in the worm caused the worm to develop in a way that was pretty crazy. If you watched the worm develop, you could see in the microscope that it was repeating early larval cell divisions over and over. And it was neglecting to transition to later developmental stages. So the animal would reach an adult stage with lots of adult parts missing because it never made those parts. And it had extra parts that are produced in the early larval stage. So I think you can imagine that this would be a really interesting mutant to study. I began studying this when I was in the lab of Bob Horvitz at MIT. What we wanted to know was, all right, there's a mutation, there must be a gene, the mutation breaks the gene must make a product, what's the product? Because if we can find that product, we can start to think about how maybe there's this clock that's telling the animal how to develop properly. And Gary Ruvkin, at the same time, was studying another gene where there were mutations that caused related and also opposite kinds of developmental defects. So that was an intriguing gene, too. Some mutations in the same gene would cause the animal to repeat larval stages, and other mutations in the exact same gene would cause the animal to skip early stages and skip to the adult. So it'd be a little animal with all these adult features, right? And missing things that we're supposed to make in the first stage. So you see, both Gary and I knew we're studying interesting genes, probably that are components of some kind of developmental clock, right? And so we were just really curious about what kind of gene products would be mediating this kind of activity. And so his lab set about to identify the DNA sequences corresponding to that other gene, Lin14. And I set about, in our lab, we set about to find the gene product for Lin4, right? And it turns out, through a lot of labor-intensive work at the time, because remember, this was early 90s, and there were no genome sequences at the time. So we depended upon information that was flowing out of the MRC laboratory in England, where John Sulston, and another Nobel Prize winner, and Alan Coulson were assembling the kind of map we needed to find these genes. And so there was a wonderful communication between the MRC laboratory and our labs, my lab, Gary Rufkin's lab, and others, where we were daily receiving information that was bringing to get into sharper focus the map of the genome, the DNA of C. elegans. And that greatly facilitated our finding the Lin4 gene. And we knew we had the Lin4 gene because that mutation that we were talking about broke the sequences of the DNA where there was a small RNA being produced. And that small RNA has come to be called the Lin4 RNA. It was the first microRNA to be identified. It's called a microRNA because it's really unexpectedly short. It was surprising that there could be enough information contained in only 22 bases or code words in the genome for this microRNA to regulate another gene so precisely. And it turns out that we now understand how that works because the microRNAs are part of a broader suite of regulatory mechanisms that were the subject of the studies that Andy Fire and Craig Mello won their Nobel Prize for, which is RNAi, RNA interference. And the important point there is there's an ancient protein called an argonaut who has evolved to latch onto these tiny sequences of nucleic acid from these genes called microRNA genes. And that flies around in the cell and finds matching sequences in the products of other genes. So what the microRNAs really end up revealing for us is a way that parts of our genome can communicate with other parts of the genome and regulate the activity of those other parts in a coherent way that enables our cells to manage the processes of development, tissue renewal, response to physiological changes, and so on. It's another layer of regulation of the gene regulatory apparatus. It's fundamental to all of us, animals, plants, and other living organisms. The microRNAs are found in plants and animals. And what else can I say? Is that good?
Like I understand
Yeah.
the science, but we have a question for
Well, was
Matt.
it comprehensible? Yeah.
Perfect.
Yeah.
Congratulations.
Thank you.
The Chancellor's reaction to the news.
Oh, I was astonished and surprised, delighted, everything you might expect. I honestly, this was not something that I expected, to be quite honest. And so, because in my opinion, the Nobel Prize to Mello and Fire encompassed all these phenomena that we study. So that was my sense, and I was comfortable with that. So as you can imagine, I was astonished by this.
I'm looking online.
Yeah. The question was, can you repeat the question? Thank you.
Thanks for giving me a purpose. The question was for people around the world who are for the first time today hearing about mRNA, what is it that you would like them to know about the science or about its potential?
Yeah, I think one of the messages, honestly, that I would hope to convey to anybody who's listening is that these kinds of studies of laboratory organisms, like in this case a nematode, Caenorhabditis elegans, are critical and key and fundamental to advancing understanding of biology. And I mean advancing understanding of biology in a way where we are constantly moving into territory that was unexpected. And I think the unexpectedness of biology is probably the most important principle, perhaps, for people to appreciate. Living things are the way they are because they evolved this way. And the processes that bring living things to the state where as we see them today, are poorly understood. And the underlying processes in our cells and the processes that manage and coordinate the action of an organism like this are still deeply mysterious. We know a lot, but in a way that can seem like an illusion, that we know so much. We all have the genome sequences, the DNA sequences, and we have identified thousands and thousands of gene products, whether they're RNA gene products, protein gene products. And so at any given moment, it feels like we know most of what we need to know, but that is actually an illusion that we have to consciously disabuse of ourselves of and leave ourselves open for the surprises.
Any other questions?
Please describe the significance of your discovery to, say, a farmer in Ohio,
A farmer?
to a layman, to a regular Joe. Yeah.
Yeah, I think the significance of this discovery of microRNAs is that it allowed us to be aware of a very complex and nuanced layer of regulation whereby genes in our cells talk to each other and coordinate their activity, and in ways that enable those cells to communicate with each other, to send and receive signals throughout the body. This is a mode of regulation of gene activity that adds to all the other layers of gene regulatory activity on the level of production of RNA products, on the level of turnover of gene products, the modifications of gene products that happen by other gene products. So the products of our genes are talking to each other in an incredibly diverse way of mechanisms, right? And the microRNAs is one of those layers, let's say, of communication amongst genes. So the importance of finding it it goes along with the importance of finding any of those other layers of gene regulation, because they all matter in different contexts, in different ways.
Yeah. The question is, on a personal level, how significant is this Nobel Prize recognition to you?
Well, yeah, I really haven't collected all my thoughts on this question. In the moment, I'm very aware that this presents an opportunity for me to speak to some of these issues that I've been trying to address, or you mentioned about the importance of public support of research, the importance of the best kinds of leadership in institutions, the importance of collegiality and a sharing of information amongst scientists. All these principles that I find so important for the advancement of science and that were so important for my success, my career. I want to shout out to the world that this is really important. All these things are important. So, the other thing is, it gives me an opportunity to mention that You know, I benefit from the people who came before me. I mentioned my parents, my father, you know, mentors along the way, you know. Ed Gruberg, my postdoc who mentored me when I was an undergraduate and taught me how to, if you don't know how to do something, walk out the door, down the hall, see if you can find somebody who knows how to do it, you know. Before you write a paper, go read all the literature, you know, something I probably wouldn't have known about. These fundamentals, you know, so you look back and see the folks who taught you the fundamentals. David Baltimore, my PhD advisor, who showed me that, you know, a simple experiment, carefully designed, strategically connected to an interesting and important question, can yield a lot, you know. David also taught me indirectly that you can have all kinds of different attributes as a human being and all kinds of shortcomings, you know, as a scientist, and it's still okay. You can contribute, right? Because David Baltimore is a man who's absolutely brilliant and inspirational, and I could look to him as somebody who I knew and knew that I was not brilliant, but I could do good work, you know, and make a contribution. So I think that one of the important messages it occurs to me to say is that, you know, often we are standing around amongst really amazing people. If we're scientists in an institution like this, everybody around us seems really amazing, you know? And often you wonder, what am I doing here? You know? And so I've come to realize that you know, I'm here because there's a few things I can do okay. There's a zillion things I can't do okay, but I work in this, you know, things get done. You know, I can work with folks who have complementary abilities, you know, and talents and stuff like that. I don't have to be the smartest person in the room. I just have to be in the right room, you know? And I think my career has been characterized by luck of being in the right room, you know?
Victor, how did you learn the news this morning?
Am I what?
How did you learn the news?
Oh, yeah, so this morning I was sleeping late, and my phone was downstairs, and we get a call from my son, who calls my wife, Candy Lee, and says, if you get a call from somebody in Sweden, answer it. Because they had called him. So I guess the poor people over there were getting desperate, and they started, let's say, all right, anybody Ambros in the general area. You know? So.
And what's it like getting that call? What do they say to you?
He said just that. Answer your phone. What did the Nobel people say? Oh, I mean, they were very nice. I mean, they said that they were really happy about this, recognizing this discovery, and they were all excited. So, you know, I took that to heart. I think they were, yeah.
Thank you. Any other reporter questions? The question is, after a few hours of the news sinking in, what's going through your mind now?
Well, quite frankly, I'm enjoying this moment right now. You know, being here with you, and I'm looking forward to later in the day getting together with Gary Ruvkin, if possible, because that'll be a fun celebration. That's what's going on right now. Yeah. Thank you.
Anybody else? Henry.
I have several questions, but there were several questions about lay from a lay perspective. What potential applications do you see this potentially leading toward in terms of therapeutics? Also, I find your shirt very interesting. You could have worn anything this morning, and you elected to wear that shirt with tigers.
Yeah.
So do tigers mean anything to you in terms of this special moment?
Okay, I can answer the second question first. This was a shirt that my wife, Rosalind Lee, bought for me a couple of months ago in a secondhand store. And it's become my favorite shirt because I get comments about it. It's the only, honest to God, I get the average of 2 comments a day from strangers. So, yeah, so it has wonderful provenance because Candy bought it, and I like to brag that it was bought at full price, which is about $5, right? Yeah. So, yeah.
And the first part of the
first
question.
is, significance of the finding, the discovery of microRNAs with regard to improving human health. Well, I mean, the most straightforward thing to say is the deeper we understand living systems, the better equipped we are to figure out what's wrong when these systems go awry in the context of disease. Yes. So thank you. So lately, I was quite astonished, actually, to realize an aspect of this issue of relevance when we attended a meeting in Copenhagen of the, it's an Argonaut meeting. So it's all about these Argonaut proteins that Craig discovered were the key partners of small RNAs, including microRNAs. And at that meeting, there was a number of cutting-edge talks that were advancing in amazing ways and astonishing ways our understanding of how this wonderful protein works, But these studies, the most amazing studies, were from people analyzing mutant proteins where the mutation in that Argonaut was identified in a family where the child has what's now called Argonaut syndrome. So these are families where the individual, the youngster, shows developmental delays of various kinds. Sometimes, complex developmental delays. It can vary a lot from patient to patient, and in every case, that patient, the child, has a spontaneous mutation in the gene for one of their Argonauts, the microRNA Argonauts. And that mutation was not inherited from the mother or the father. It's a spontaneous mutation. And it turns out these mutations are in amino acids of Argonaut that are conserved in C. elegans. So our lab and these other labs had started to take those same mutations that were found in the human families and put them into the Argonaut of C. elegans and try to learn what we can about how this protein is broken, And as I say, several labs reported really amazing findings that, on the one hand, were telling us giving us new insight into how this amazing protein, Argonaut, works. So the basic sciences in the audience were learning from the mutations that were presented to us by the families. And several families were there at the meeting, and several of the children were there, and we got to meet them. And for me, this was an amazing experience because I was, for the first time, trying to process this idea that we basic scientists are learning from these patients, right? And then there was really interesting discussions at the meeting, of course, about how we basic scientists could end up somehow helping these families, if not now, in the future. And so there was talk about potential therapeutics, but the discussions were actually collaborative discussions between the patients, families, and the scientists in the room, you know. So it was I think that, I guess it's a long-winded way of saying that the significance of this kind of work to human health, to me, is probably, it's unexpected. Of course, there's companies out there, obviously, who are using small RNAs as therapeutics, and there's a big thrust here, and amongst those small RNAs are microRNAs. So it's a very active area of therapeutic development in industry and in academia. But I wanted to tell you that story because it was so personal and so interesting for me. And it actually, then you start to think about how can I shape my research in a way that I can continue to, uncover fundamental things if I can, but also to try to think and along this other angle at the same time.
More questions? If not, why don't we get our two Nobel
Oscar
laureates?
has a question.
Oh, I'm sorry. Okay.
Victor, do you have anything to say to the science trainees or?
What to say to the science trainees or younger generation? So those, if any of you are in the room who are science trainees or younger generation, you know that I can really become a flamer sometime and really, blather on and on about, the basic thrust of it is, you guys, each one of you is talented. Each one of you has something to offer, not just something really special and important to offer, and the field that we study, let's say RNA biology, if we could put it that way, is a field that's just incredibly exciting right now and will continue to be, indefinitely. So keep up the good work and just know that you are the right person for the job you're doing, and you're going to do great things.
Thank you. If you could please thank your son for calling you. No one's happier than we are that you're here. So let's get our two Nobel laureates together. Craig Mello, co-recipient of the 2006 Nobel Prize.
Sure. What a long overdue and wonderful day it is. They found a way to surprise both of us because I was surprised that they had awarded the prize to us, skipping over Gary and Victor for now almost 18 years or so. So it's kind of a relief, to finally feel like justice has been done. The discovery that Victor and Gary made was an inspiration to me. Also, of course, Victor was my mentor, and Gary was on my thesis committee. So I knew them both very, very well, and they both inspired me for different reasons, just full of enthusiasm and curiosity, really believing in this little organism and what it had to teach us, and being right there with advice along the way. Victor's just a tremendous mentor, and I had the pleasure of finishing my PhD in his lab. And without his help, I don't think I wouldn't be here without Victor. Victor really made it happen, believed in me enough to help me get through a really difficult time in my own career when we were trying really hard to get, micro-injection of genetic information back into the animal to work, and it didn't work. But with Victor's help, we really got it working. And that's sort of, so I feel really emotional right now to be here to celebrate Victor. Just an incredible, inspiring person. He's the kind of person who always has an insightful question at every single meeting that you go to. He sparks all kinds of ideas constantly. I also want to mention that Victor was a co-founder of the UMass RNA Therapeutics Institute, which really, again, we owe great thanks to both the Nobel Committee and to the state of Massachusetts for investing in our institution. After the 2006 Nobel Prize, the state came up with $100 million that helped make this building possible. And in so doing, also created the foundation for an RNA Therapeutics Institute, which would build on the expertise in RNA biology that Alan Jacobson and others, Melissa Moore, Phil Zamore, Michael Green, had established here at this institution. And so it was extremely timely that that institute was named the RNA Therapeutics Institute. At the time, there were no RNA therapeutics, but it was a clear aspiration. And again, Victor is really too humble he should take credit for the therapeutics because they use his mechanism. The therapeutics actually that are made today are short pieces of genetic information, just like the one he discovered first, Lin4. And we can now make these, we call them search queries because they program a search engine, which is this Argonaut protein. It's the Google of the cell. And for those in the lay audience out there, imagine these things can search inside of our cell and find matching information. And they do it at 100 million collisions per second. They're incredibly efficient. They're really fast. Our body is doing computations all the time. And we know like really this much about how organisms handle information, and yet it is fundamentally important to human health, human biology, and the future agriculture. I mean, there's so many applications for improved understanding of how our body handles information. And everybody knows about DNA, and I guess the whole world got a crash course in RNA. Now, sometimes Victor will get this too. I've been given credit for discovering mRNA. You will get it. mRNA is different, okay? These little RNAs, in a way, have much better therapeutic potential because they're tiny, they're easy to make. They don't need to be put into a compartment and delivered inside of a shell. They can be delivered directly. And one of the great things that Victor and I and others here have been able to do is recruit some really amazing RNA chemists who have been helping us and the community make these into drugs that are more effective and deliver now really throughout the body. So you guys, the lay people out there, more and more of you are going to be getting these kinds of medicines, and we can only hope that they will not only help people with Argonaut syndrome, which I think they can actually, and many other diseases, even cancer, Alzheimer's, pretty much you name it, there's a genetic basis for every disease. And so thanks to the work of Victor and Gary, all of this is now possible. I mean, really, they've just been, at the forefront of a really, really exciting revolution in biology. It's one where, pretty much, as Victor said, we just keep realizing how, with every new discovery, we just keep realizing how little we really understand the nature of how we work and how much more work there is to do. So I'll stop there, and I'd just encourage any young people, if you're inspired by the work of Victor and Gary, get involved in biology. Biology is having an amazing revolution, and so is medicine. They really go hand in hand now. So anyway, one more round of applause for Victor and Candy.
Do we have any other questions for our laureates before we move on to the rest of the day's activities? One more? What inspired you to go into this field?
What inspired me to go into this field and what was my initial expectations? Well, I mean, what actually happened was I was a postdoc in Bob Horvitz's lab, and I was basically saying, hey, Bob, what do you think I should do? And Bob says, there's this mutation that looks really interesting. You know, I think you should try to figure it out. And so that's got me into the field. And, but of course I was immediately captivated by the nature of the mutation in this worm. As I mentioned earlier, it looked like it had a defective developmental clock. So,
What year was that?
let me see, I graduated in '79. It'd be 1979, yeah. Does that make sense, Candy? Yeah. Yeah, I started in his lab in '79. Yeah. So expectations at the time were, I think you can tell it, I did not expect it to be anything necessarily to do with RNA biology or small RNAs or anything. The expectation was, frankly, I hope this is a project that can get me some publications so I can eventually get a job, you know. So, yeah. So honestly, it was, yeah, that was my expectations. Whether it would, you know, break new ground scientifically, that was beyond any expectations. Oh, the surprises along the way and the biggest obstacles? I would say the biggest surprise was when, you know, Candy, Rosalind, and Rhonda were identifying the gene product of LIN4, and we were repeatedly stymied in trying to test various hypotheses. Oh, it must be this protein that's encoded nearby. No, you know. Oh, maybe it's a very unusual protein coming from a tiny RNA. And then they were able to show, no, it's not, can't be the case, you know. Oh, and then could it be that really tiny RNA at the bottom of the gel? Those of you who run gels will know sometimes there's all this schmutz at the bottom of the gel, what we call it, you know. And it turned out that was the small RNA that we're all talking about, was something so unexpected that we had kind of ignored it for a while as just, you know, schmutz. So there were constant surprises, yeah.
Any final questions? Okay, so for the members of the media in the room, we will be posting new information, updates, photos, all kinds of resources on umassmed.edu/news all day. So that's a good place if you're looking for additional information as time goes on. You can find us on social media at UMass Chan. And we're hoping to get the two of you together this afternoon. We're still working on that. So thank you so much for coming out and for hearing from our now second Nobel laureate here at UMass Chan Medical School. Thank you.